for giving me the opportunity to speak today. It's such a wonderful event. It's a real honor. I'm dressed as myself in 40 years when hopefully I'll have an established lab. For the moment, however, I'm going to present to you my postdoctoral work, which I've done in the Fromke lab at NYU. And by the way, greetings from New York. It's still dark outside, early morning. I'm going to speak to you about the fourth age, what I call the age of the soldier moms. Let me know if my slides are changing. All right. When we first hear the word soldier, we might picture a classical battlefield scene, something from the history books. However, for most of us, our everyday battlefield looks something rather than this. Especially now in times of pandemic, our daily fights involve feeding, dressing, and schooling our kids. Parents are constantly fighting. And it seems that now more than ever, the expectations about new parents are extremely high. Here is how we see parents. These are different words extracted from texts. In black, you can see words which are used to describe soldiers as well as parents, and in red, words used for parents mostly. And I hope that I can at least partially convince you that the way we look at parents is we consider them as superheroes. They are indeed soldiers. So for the rest of the talk, we're going to dig a little bit into neuromechanisms which confirm some of the superpowers of parents. We're going to mainly talk about mothers because in most of the mammalian species, mothers are the primary caregivers. So new moms have to learn quite rapidly how to provide food, protection, and care to the offspring. We already heard a lot about oxytocin, and this is indeed because the neurocircuits involved into the food supply, which is the lactation, are very well described. And I hope that for the rest of the talk, I can actually convince you that oxytocin is also involved in other sorts of, like, broadly maternal care and not only lactation. What we know about oxytocin is that it is synthesized and released from the maternal hypothalamus in the brain. It gets into the bloodstream to facilitate the milk ejection, and this happens during nursing. However, sometimes oxytocin release and milk letdown can happen in the absence of a nursing infant. And this has been described in humans. So curiously, human mothers describe that they can experience spontaneous milk letdown only by hearing the sound of a baby cry. And here I'm showing you a very fascinating study where they put human moms in the scan and they played baby cries to them. To us, this might seem as an ultimate torture, but what is important is that you can see the human hypothalamic region here, the blue arrow, is actually activated during baby cries. And this activation, most importantly, is strong enough to trigger oxytocin release in the blood. You can see the plasma oxytocin levels peak several minutes after moms hear the baby cries. So that tells us that infant vocalizations are extremely powerful stimulus to recruit neurocircuits classically involved in nursing. However, how that happens is something that we're going to try to tackle for the rest of the talk. Are moms crazy, or there is some sort of a biological substrate which can activate hypothalamus neurons via auditory cues? Let's see. In our lab, we work on mice, and mice are very curious because they exhibit this very stereotypic pattern of behavior known as pup retrieval in response to vocalizations from pups. What happens is that when a mouse pup is isolated from the nest, it will cry, it emits these ultrasound pup distress calls. You can see the spectrogram here. They will attract the mom, she'll pick the pup and bring it back to the nest to safety. And this is very similar to human moms, which have this behavior to pick up and hold their baby if they're crying. So we decided to record from oxytocin neurons in mice. In maternal mice, we also call them dams. Here I'm showing you what it looks like, the mouse hypothalamus. This is the paraventricular nucleus of the hypothalamus, this heart-shaped structure, called also the PVN. The way we record from those neurons is that they're tagged with channel rhodopsin. So if we insert an optical fiber connected to a blue laser in our electrode, we can actually recognize oxytocin neurons because they will emit spikes in response to blue light pulses. And the rest of the neurons in the brain, they will remain silent. What we do is to record vocalizations from isolated pups, loop them in a circuit, and play some sort of a podcast to the moms. It's a very similar torture to the one we've seen with human mothers in the scan. So here's what we saw. Very strikingly, we found that oxytocin neurons drastically increase their neuroactivity after we present pup distress calls. And even more surprisingly is the fact that other cells do not modulate their activity, telling us that there is some sort of a cell type specificity about the processing of these infant vocalizations in the maternal hypothalamus. So oxytocin cells are specifically tuned to that. So we decided to look at what brain areas might relay auditory information to the hypothalamus. So we injected Cre-dependent rabies virus in the PVN. So these are virus which will infect all hypothalamic oxytocin neurons, will jump one synapse backwards and will stain all area projecting to them. So we specifically looked for auditory areas, and we found that not very well studied actually part of the auditory thalamus called the posterior intralaminar thalamus, the PIL, projects to these oxytocin cells. So we decided to record in the PIL, and we saw that indeed these thalamic neurons respond to the pup distress calls. Then I'm not going to get really into details, but those of you who are interested in the circuitry is that, so we found that thalamus receives main input from the inferior colliculus and less input from the corticofugal projections from auditory cortex. So now the big question here is how the thalamic discharge during the pup calls might trigger this increased activity in oxytocin cells. So to tackle that, we went to the experiments in brain slices. So what we did was to optogenetically stimulate thalamic fibers in the hypothalamus while we record from oxytocin cells. And we saw that while synaptic excitation basically stays the same, synaptic inhibition drastically decreases after we stimulate the thalamus. And this leads to this decreased the inhibitory to excitatory ratio. So we think that this decrease in inhibition or the disinhibition of oxytocin neurons can actually lead to this increased output of these cells, so they can now spike and actually release oxytocin in downstream areas. But first we wanted to check what are the synaptic mechanisms. Very briefly, this is actually triggered by internalization of GABA receptors, for those of you who are interested in synaptic physiology. So what is important of this thalamus to hypothalamus pathway in maternal behavior? Well, if we inject inhibitory dreads in the thalamus, we can actually inhibit those cells with this drug called CNO. So we decided to test pup retrieval behavior, and what we saw was that in moms which had their thalamus inhibited, even though they retrieved the same amount of pups, they did that way slower. So they kind of looked not very motivated to do so. So we thought that, well, maybe this thalamus to hypothalamus pathway is important to trigger oxytocin release in motivational areas. So then when we went to look in the ventral tegmental area, the VTA, we expressed genetically encoded oxytocin sensor there, developed by the Kwon lab at Johns Hopkins. So this oxytocin sensor is actually able to detect oxytocin release, but it needs to be activated by blue light. So we also put this optical fiber in the VTA. What we do is that we expose our moms to listening of pup calls in the presence of blue light, and then we look in VTA neurons. And indeed, we found, you can see here, actually everything which is yellow are cells that have detected oxytocin release, which happened during the pup calls. Here is briefly what I've showed you is that it seems that hypothalamic oxytocin neurons do respond to vocalizations from pups via this non-canonical auditory pathway. And this is important for oxytocin release in motivational areas and also important for speeding up retrieval behavior. What can be the physiological relevance here? So it seems that these so-called nursing circuits can actually be recruited by auditory cues as well, and not only by somatic touch during nursing. And that release of oxytocin is important for maternal care broadly and not only lactation. What might be the benefits for infants then? Well, they can actually attract maternal attention by exploiting different signals. So not only by somatic touch, but also by emitting auditory signals. And you can imagine that this can expand to other cues as well, such as olfactory and even more visual if we speak about humans. By that, I'll end up here. I want to thank my entire lab, Rob Fromke, who is an amazing mentor, NYU, and also my collaborators from outside, my funding sources, and of course you for your attention. And I'm more than happy to take questions.
Thank you, Silvana.